Really?
Okay, it looks like everyone's here, so let's go ahead and start. So we're now talking about microbial growth. So we've gone through nutrition, seen how they've taken up nutrients. Now what are they doing with them? So how do they grow and how do they divide? So bacterial cell division is not as extensively studied as mitosis, say, in eukaryotic cells. There's still a lot of people who make their careers studying this. But I want to just go over some of the basics of bacterial cell division, at least in terms of the general principles, because each bacterial species or different genera or different phyla probably has different strategies of doing this. But for the most part, bacteria, whether they're coccoid or bacillary or whatever shape, tend to almost always divide at the exact midpoint. And they do that using an analog of a well-known eukaryotic cytoskeletal protein, tubulin. The analog in bacteria is a protein known as FTSZ, or FTSE, as it's more frequently referred to. FTSE will form, along with other proteins, a very complex structure at the exact midpoint of a cell and will actually constrict. So it will depolymerize to form a septum at the midpoint of the cell. So the FTSE ring is actually contractile. And what you're seeing here in this these staphylococcal cells that are dividing are septa that are forming that will eventually pinch off and divide those cells into two perfectly spherical half cells. Here's a better series of electron micrographs of Staph aureus, where you can see on the left in panel A the earliest stages of septum formation. So you can't see the FTSZ or FTSE ring, but the FTSE ring has formed at the exact midpoint of this coccoid-shaped cell and is beginning to contract. It's beginning to pull in on itself. So it's synthesizing new cell wall material. So new peptidoglycan is being synthesized here, as well as new cytoplasmic membrane. Then eventually, as it's shown here in B, the septum is formed completely, and then eventually the cells are going to separate. So that in any cell, you always have, even in a coccoid cell, you actually have poles, referred to as the new and the old poles. And there are some morphologic differences in terms of protein localization, where some proteins will localize to the old poles of the cells, which would be shown in these two corners, the northeast corner and the southwest corner. Whereas where this cell has divided and the peptidoglycan is the freshest, it's the newest, the cytoplasmic membrane has been synthesized there most recently. That's what's referred to as the new pole. We won't go into much of the ramifications of old versus new poles, but know that there are differences in how proteins localize to those poles. Now, how is it that bacteria determine precisely the exact midpoint of the cell? And they do it in a really fascinating manner. One that's fairly well studied is a complex of two proteins known as min-C and min-D. So we just call them min-C-D. This is in essence a protein metronome. So any of you who are musically inclined know what a metronome is. It's that thing that helps keep time on a piano, where you have an arm that slowly moves to the right, slows and stops at the right, starts the progression back to the left, accelerates, gets to the left, pauses, and changes directions. Bacteria have actually evolved a protein version of that metronome this min-CD protein. Now, unlike a metronome, the min-CD is not about timing. We use metronomes to help keep time in music, so I'm told. But this is all about determining the precise midpoint of the cell. So what's shown in this series of micrographs is green fluorescent protein tagged min-CD over time in the same cell. These numbers reflect seconds. So this occurs quite rapidly in this rod-shaped bacterium. You can see a DIC, or just an unstained version of this same bacteria in the lower right. So you can see the rod-shaped bacteria and where the poles are. I don't know actually why one pole is dark here and the other is light. I'm not sure what the meaning of that is. But if you watch the movement over time of min-CD, you see that starting at time 0, you have in this northwest pole of this rod-shaped bacterium the collection of min-CD. At 4 seconds, it's still there, perhaps even more concentrated. And at 9 seconds, it's still in that pole. But then 4 seconds later, you don't really capture the min-CD in between there. You see that it's already moved to the other pole, the opposite pole, and is beginning to coalesce for another portion of time. So you see out through 31 seconds, there is a coalescence of min-CD at the opposite pole. Then again, within 4 seconds, so recognize the timing here is actually kind of important, because look at the amount of time in seconds that this protein complex spends at that pole. So anywhere from 13 seconds to 31 seconds, you can still see that protein complexing in that pole. Within 4 seconds, it has disappeared from that pole and is now coalescing again in the opposite pole, where it will stay from 35 seconds to roughly 48 seconds. And then again, in the span of about 4 seconds, it's gone from that pole, coalesces at the other end. So you can see that this protein is actually spending, or this complex of proteins is actually spending more of its time at the poles than at the midpoint. And that's the important point. The FITZ ring, that polymer of proteins, of the protein FITZ, the polymerization is actually inhibited by min-CD. Wherever min-CD is, FITZ is much less likely to polymerize. And so if you look at the localization of this protein over time, it spends the least amount of its time at the equator of the cell. It actually, once it starts to move from one pole to the other, it moves very rapidly to the other pole, where it recoalesces, polymerizes, depolymerizes, and moves to the other end. So it's that inhibition of the FITZ ring by min-CD which allows bacteria to select always the precise midpoint of that cell. So that's where the that's where the FITZ ring will form. And what actually guides that min-CD protein complex is something that we've already talked about, the bacterial analog of actin, MREB. That cytoskeletal protein that's responsible for, especially in rod-shaped and spiral-shaped bacteria, that actin analog that's responsible for cell shape, that forms these arcs or coils in the cell. These are actually the tracts that min-CD follows to go from one end to the other end. Without these proteins, min-CD doesn't get localized quite as well to one end or the other. So everything kind of ties back to the cytoskeleton. Now, once that FITZ ring forms, it's not FITZ by itself. It's actually a complex structure called the divosome. The divosome is made up of a lot of different proteins, and quite honestly, some of which people still don't know what they do. There are quite a number of people doing research in microbiology who have made their entire careers studying how it is that the divosome will divide cells. But we know that in addition to this polymer of FITZ proteins, which has the ability to contract, and it does that contraction by hydrolyzing GTP into GDP, so it's a G protein. It also is associated with other proteins that will, among other things, make certain that at the site of that Z ring, which is going to be the site of the new septum formation, that peptidoglycan synthesis is maximized at that point. And this makes sense because the septum that's being formed between two nascent cells has to have more cell wall material synthesized. So proteins like this FTSI, you can see that this protein communicates information between the cytoplasm of the cell and the periplasm in the case of this gram-negative bacterium. And so this helps to make certain that as peptidoglycan is remodeled by autolysins and then resynthesized or remade slightly larger using the things like transglycosylases and the transpeptidases, the things that you're studying about for the exam this week, that's being directed by this divisome, this really complex series of multiple proteins that form at points around the Fitz-Z ring. So in this cutaway shot of a dividing bacterium, you can see the entire Fitz-Z ring goes all the way around the circumference of the cell, at least as far as you can see. And then you can see various divisomes formed at various points around that circumference, each one of which is now dictating peptidoglycan synthesis on the other side of the membrane, so that as GTP is hydrolyzed and this Fitz-Z ring depolymerizes, in effect, think of a coin purse when you pull the strings tight on something and things pinch together. So as that's occurring, new peptidoglycan is synthesized. And in these in these micrographs at the bottom, you can see, especially in the blue and in the red, the blue time series is staining DNA. So you can see the partitioning of the DNA in these bacteria. But the Fitz-Z is actually now shown here in red. And you can see the formation over time, starting from a cell on the left-hand side, the left lower corner here, that really doesn't have a septum yet. And then over time, you see that the Fitz-Z becomes more and more localized to that midpoint until eventually the septum is formed, always at the precise midpoint. Now, the reason that most of these genes are named FTS is kind of a historical problem, and problem is not the right word, but a historical fact, is that because as people doing early genetics in bacteria realized that they could make mutations in genes that they had no idea what they did, that a lot of these genes grew as filaments. Rather than dividing into cells at the precise midpoint, certain mutations would make cells filamentous. So these filamentous temperature-sensitive mutants, that name still dominates many of the proteins involved in the division of cells. Okay, that's all I really want to say about the division of bacteria. We could probably talk more about it, but I don't know much more about it, so I guess we'll stop at that point. And we'll come back and talk about a topic that I broached a little bit on Friday, the growth curve of bacteria. And so recognize that I don't really like teaching about this growth curve, but it's kind of one of the things I feel like I have to do in a microbiology class, because this growth curve, as I'm showing it to you here, is entirely a laboratory artifact. But we do have to deal with microbiology in the laboratory as well as in the natural environment. So we'll proceed. And I want to talk about how it is that microbes or the populations of microbes differ during each one of these phases as they progress in the laboratory. So lag phase, that initial phase here early in time where there is no increase in the number. So the y-axis here is the number of viable cells. But during this lag phase, there is no increase in cells. This is usually because of the case that when we inoculate a new culture in the laboratory, we're usually doing it with a culture that we started the day before, that's grown in the incubator, that's probably out here in stationary phase, possibly even into the death phase, but most certainly in the stationary phase in the case of E. coli, our favorite lab rat. So if you take bacteria from the stationary phase and inoculate it into new media, it goes through this period where it just doesn't know what to do. It's gotten used to living in very noxious conditions where it's been living in its own waste product for a period of time in a stationary phase. So it goes into this lag phase where it really can't yet start to divide. But it's still metabolically active and it's adapting to that new environment during the lag phase. So during the lag phase, there is no increase in the population. So the number of cells remains relatively constant. The cells actually may and do change in size. So as they get ready to divide into two, remember that bacterial cells actually do increase in size before they divide. So there is cell growth. I put it in quotation marks here because typically when microbiologists refer to cell growth, they're talking about population growth. In this particular instance, I'm talking about the growth in physical diameter and physical length of an individual bacterial cell, not a population. So increase in size of individual cells. During the lag phase, this is what's often referred to as unbalanced growth. It's unbalanced or referred to as unbalanced because not all of the cellular constituents are being produced at the same rate. So these bacteria in the lag phase, in effect, are becoming prepared to grow in this new media that they found themselves in. So at some things are going to occur more rapidly. So in lag phase, protein synthesis is going to exceed DNA synthesis. Because before the bacterium can divide in this new environment, it has to first produce the enzymes necessary for division. If we started this culture with some cells from the stationary phase, the most common thing that we would do, many of those cells are not thinking about, I shouldn't speak teleologically, but they're not physically prepared to do reproduction at a very high rate. So they have to resynthesize the proteins necessary to synthesize DNA, to divide DNA. They need to synthesize proteins for metabolic pathways that they need for active transport. So they're just preparing for growth. So protein synthesis occurs more rapidly than DNA or phospholipid or peptidoglycan synthesis. It's making all of the enzymes necessary for when it finally decides to start growing. So some of this increased protein synthesis is for nutrient uptake. So the bacteria, as they were living in a nutrient-challenged environment in the stationary phase, stopped producing some of the active transport proteins because there was nothing there to transport. So saving their energy trying to survive until they found themselves in a new environment, they don't have all of the machinery necessary to take up all of the delicious nutrients that they find themselves in during the lag phase. So they have to resynthesize those products as well. And this is all in preparation for the bacteria just going absolutely wild and dividing as rapidly as they can. That's what's known as the exponential phase of growth. So in exponential phase, this is what's referred to as balanced growth. All of the cell constituents are growing now. DNA is being replicated, RNA is being synthesized, new protein is being made and turned over. These cells are, as healthy a population as you can imagine. So think of these phases not in terms of individual cells, but in terms of the population. During the exponential phase, virtually all of the cells are physiologically at the exact same state. They've gone through lag phase together. They now have unlimited, as far as they can tell, nutrients. Toxic byproducts have not yet accumulated to the point where they're a detriment to growth. So these bacteria are growing as fast as the conditions will allow them to do, and all of the cells are doing that, the entire population. These cells are the healthiest. These are the cells that microbiologists will always use to do biochemical or physiological studies. It makes sense because the cells in that population are relatively uniform in terms of their physiology at that point. So that's why people use them at this healthiest point. I personally think that's perhaps not most reflective of how these cells live most of their lives, but I understand why we have to do this during the exponential phase. So again, as I mentioned, during the exponential phase, nutrients aren't growth limiting, metabolic waste products are not limiting either, but obviously this can't go on forever. Eventually the bacteria in this exponential phase are going to you know, not like living in their own excrement. They're going to start, you know, resources are going to become limiting, and they're going to slow their growth and go into stationary phase. Me personally, I am a big fan of the stationary phase because I think that in terms of most microbes in their natural environments, outside of the laboratory, which is where I prefer to think of bacteria in their natural environment, probably are living at this balanced level where in stationary phase, the number of cells that are dying is relatively equal to the number of cells that are dividing. So you see that cell population doesn't change much over time during stationary phase. It's not that cells are not dividing, they are, but an equal number are dying. So this is part of the reason why most people don't do experiments with stationary phase bacteria, because you don't have a uniform population anymore. You have some populations that are, in effect, dead. And remember that in microbiology, we refer to dead microbes simply as those cells that can no longer divide. It doesn't mean that they're not physiologically active. It doesn't mean that they can't produce toxins if you're thinking about pathogenic bacteria. They've just lost the ability to divide. That defines death in microbiology. And if you think about, take this to the absurd level, one E. coli cell, which in the laboratory will generate every 20 minutes, in exponential phase for 48 hours would give a population 4,000 times the weight of the Earth. So small bacteria, but doubling every 20 minutes could get really large. Obviously, this can't happen. So this stationary phase occurs in virtually any environment, whether it's in the laboratory, in a broth culture, or in Lake Matoka, or in our intestinal tracts. Wherever it is, the bacteria eventually reach a point where growth is just not a fun thing to do, not an easy thing to do. I guess it's still fun, but it's not an easy thing to do. So stationary phase arises because of nutrient depletion and or the accumulation of toxic metabolites. These stationary phase cells, this entire population, goes through an adaptation. So it changes its whole gene expression pattern in order to survive these trying times. So it's a completely different physiology that's going on during stationary phase as compared to exponential or lag phase. They're actually capable now of coping with this environment. So some of the things that they do to cope is they decrease the synthesis of their replication enzymes, because better to try and survive, pull up whatever nutrients that you can, and maybe divide more slowly. So they'll decrease replication enzymes, they'll decrease nutrient uptake proteins, because it's quite simply the nutrients aren't there in order to be transported. So perhaps they have to transport new products. So oftentimes the breakdown products of their brethren bacteria, because some of the bacteria are dying, lysing, being released into the environment, and bacteria don't mind being cannibals if they have to be. They really don't, they don't give it a second thought. But those are different forms of nutrients oftentimes. So they're taking up different things. But these bacteria are actually, at least in terms of how I think about microbes, in terms of association with the human or animal microcosm, these microbes are tougher to kill. These are microbes that are now adapted to a difficult lifestyle, which is what really we're trying to create for microbes in our, in terms of pathogens. We don't want them to be in exponential phase, to grow really rapidly. So these bacteria become actually much more resistant to antibiotics. So it becomes more difficult to kill them from a clinical point of view with antimicrobials. And stationary phase, as I keep harping on, is probably much more reflective of the natural situation for most microbes. But then, of course, in the laboratory, or perhaps in other environments, as well, if the growth continues or if time continues without any addition of additional resources or taking out some of the metabolic byproducts, perhaps by dilution or something like that, the cells are going to enter death phase. Very, very logical. So the continued incubation leads to death of cells. This doesn't mean during the death phase that there aren't cells that are reproducing. There are. It's just that now the balance is more towards cells losing the ability to replicate as opposed to actually replicating. But there are populations of cells within that that are perfectly happy to cannibalize the dead remains, the guts of bacteria that have died before them in lice, and will still be able to divide. So there is still a viable population within this death phase. Now, there's a really interesting concept that's actually still hotly debated in the microbiology field, and it has a lot of public health implications, and that's a state that bacteria can be in where microbiologists consider them dead because you can't grow them in the laboratory. Yet, you can look by various staining mechanisms and see that physiology is still occurring within those cells. There are still metabolic pathways that are active within those cells. So they're still living in some way, but you can't grow them in the laboratory. So they're viable but non-cultivatable. VBNC. Why this is really a matter of public health interest is that there are a lot of people, myself included, who believe that simply not being able to cultivate these microbes in the laboratory, a highly artificial environment, is not really reflective of what these organisms might be able to do. There are a large number of people who believe that these viable but non-cultivatable state of microbes could actually be a major means of transmission of microbes, even pathogens, from host to host. And from a public health point of view, this is a nightmare, because from a public health point of view in the laboratory, you can't cultivate these things. They're coming out perhaps, the most common way to think about this is fecal contamination of water or food supplies. And so if you're basing your tests on whether the hamburger is contaminated with bacteria that could cause Shigella or salmonellosis, if that test is based on the ability to grow those bacteria, if they're in this VBNC state, then you're going to get a negative test. And yet these microbes may yet be able to be revived in a natural situation, so when they gain access to a new host. So this is a major worry of public health microbiologists, this viable but non-culturable state. Okay, so there, I did my duty. I walked through the growth curve. You can tell anybody that asks you about microbiology that I actually did it. You all now know about what you have to know. Let's talk about something only slightly more interesting, at least to my point of view, about environmental conditions and how they affect microbial growth. It is a lot more interesting, really. We could probably spend an entire course in environmental microbiology talking about how microbes in the environment are affected by things like acidity or alkalinity, osmolarity, oxygen pressure, things like this. So you can all imagine, because you and I, our growth and our health is affected by things like atmospheric pressure and osmolarity, that microbes are no different in that way than us. They are affected, their growth is affected by a lot of different environmental conditions. I put this down kind of tongue-in-cheek that temperature is most important. It's really not the most important. It's just the one that I've chosen to talk about, so that makes it the most important. We're going to go through the effects of temperatures on various microbes. It is important for a lot of environmental microbiology concerns, but also industrial microbiology concerns. So a lot of things that we can do industrially in terms of bioremediation, in terms of the production of things for the pharmaceutical trade, are actually done by using microbes that behave well at high temperatures. So temperature is very, very important. And so as we talk about temperature as an environmental condition for bacteria, the three temperatures, the cardinal temperatures that are important to remember for any bacterial species are the minimum, the optimum, and the maximum species. And why this is particularly interesting for bacteria is that bacteria and archaea, unlike eukaryotes like you and I, have got a huge range of temperatures at which they can not only survive, but in fact thrive. You and I don't do very well, nor do any of our eukaryotic brethren, do very well if the temperature is higher than, like, say, 50 degrees centigrade. I don't even know how well we'd survive at 50 degrees centigrade. But prokaryotic species survive and thrive at much higher, and lower temperatures than most eukaryotes. So this is a very noisy table, but if you just take a look at it, one of the things that you'll see is that in terms of minimal temperatures that microbes can live at, or they can get down to below freezing, below 0 centigrade. So there are environments, especially saline environments, where water doesn't freeze at the typical 0 degrees centigrade that we think of. So these are environments that certain microbes, like this Bacillus cyclophilus, can grow in at very low temperatures. But more interesting to me, and therefore more interesting to you, is as you come down on this list of non-photosynthetic prokaryotes, you get down into microbes where you're looking at the optimal temperature and it's going up as you're going down here. So you'll see that most of the bacteria that you've heard of, that you're concerned about, like Neisseria gonorrhea, E. coli, staphylococci, have, not surprisingly, evolved to have an optimal temperature that's the same as your temperature and my body temperature, because these are microbes that are adapted to live at that particular temperature. But if you start looking at environmental bacteria, you can see Bacillus stereothermophilus has an optimal temperature of 60 to 65. The only microbe that I want you to remember from this table, and we're going to come back and talk about it again because it's a critically important one, is this one, Thermus aquaticus. Thermus aquaticus is a bacterium that we'll talk about maybe later in this lecture, certainly by Wednesday. It has an optimal growth temperature of 70 to 72 degrees centigrade. That's damn hot. You can't keep your hand in water that hot. You'll get scalded by water that hot. Yet that's the optimal temperature for replication of that bacterium. I'll give you a hint as to why this is really important for any of you who have ever done polymerase chain reactions. You recognize the fact that that extension temperature, we'll talk about PCR, but for those of you who understand what PCR is already, that extension temperature that you do, the 72 degrees, the third step of a thermal cycling reaction, that 72 degrees is not accidentally chosen, it's not arbitrarily chosen. It's the DNA polymerase from this bacterium, Thermus aquaticus, discovered in a hot spring in Yellowstone National Park, growing optimally at 72 degrees. We have actually robbed that bacterium, cloned the DNA polymerase from Thermus aquaticus, Taq polymerase. That's where the word Taq polymerase comes from. This has revolutionized molecular biology. So this is part of the other reason why we talk so much about the temperature range that microbes can exist at. But so Thermus aquaticus, really important. Remember this one, and we're going to come back and talk about how PCR works. But there are microbes that go even higher than that. If you look at any of the microbes that in this particular list that have higher optimal temperatures, they're no longer bacteria. These are archaea. Remember, the archaea are that really old domain that probably dominated Earth when Earth was hellaciously hot. Well, as the Earth is cooled, these microbes have probably gone through ecological compression. They probably have much fewer environments where they can live, but they're still dominant at areas such as deep sea hydrothermal vents, where temperatures can be really hellaciously hot. There are archaea that actually have the capability of replicating at 13 degrees above boiling temperature. So our proteins would long before that been denatured and completely just taken apart, destroyed at this temperature. Yet the optimal temperature for growth is 6 degrees above boiling temperature. So this is just amazing. Among prokaryotes, you can also look at photosynthetic bacteria. Photosynthetic bacteria, because of the complexity of the photosystem and all of the internal thylakoid membranes that they have, don't do as well at high temperatures. There are some photosynthetic prokaryotes that do well at even as high as 79 or 80 degrees centigrade, but for the most part, most photosynthetic bacteria, like most photosynthetic eukaryotes, don't do very well at high temperatures. But low temperatures are a different matter. So while microbes really can live very well and thrive very well at low temperatures, there are some eukaryotes that can as well. My favorite one among the eukaryotic algae is Chlamydomonas nivalis. This is an alga species that has as its optimal growth temperature 0 degrees centigrade. It can actually replicate as cold as minus 36 degrees. It dies much above 4 degrees. So this is a very highly cold adapted algae, and it's a beautiful algae. So any of you who have ever been to the Sierra Nevadas in California may have seen this, that especially as snowdrifts begin to melt, this Chlamydomonas nivalis produces a dispersal spore which is red. I don't know why it's red, but the reason why I love this is I'd seen this picture for years, but back about 10, 12, no, actually it was longer ago than that, 2002, Ann and I were on vacation at Lassen Volcanic National Park, and we were hiking at about 9,000 feet, and we found this snowbank that was even more brilliant than this. It was beautiful pink. Just imagine like a snowbank, probably as big as this auditorium, and it's just bright, bright pink. We were having pink snowball fights. We were making pink snow angels. I was just loving this. And then all of a sudden, to hear Ann tell the story, when the next group of hikers came along with their children, I don't believe this is true, launched into professor mode and started giving them a lecture when they asked about the pink snow. She claims that I actually used the term psychrophilic eukaryotic alga to these tourists. And she said that they couldn't get away from us fast enough. I think she exaggerates. But this stuff is absolutely gorgeous. It was just so much fun to play in that. Okay, but in terms of an individual bacterial species, regardless of what temperature it uses as its optimum, there are minimum and maximum temperatures, and there's reasons for there to be minimum and maximum temperatures. So if you look at the growth rate of any bacterial species, here on the y-axis, with growth rate increasing at the top and temperature increasing on the x-axis, you'll see that while there's an optimal temperature here, so this would be the favorite temperature of the bacterium, if you look at there's always a minimum temperature here, and if you think about it, it's very obvious, or it's very easy to think of the reasons for a minimum temperature. Probably the main thing is that the membrane gels. Any of you who have ever stuck a chicken that was cooked, or turkey, or maybe even any other kind of meat in the refrigerator and seen that the fat that is dripped off, it turns into gelatin, realize the fact that fatty acids and lipids at cold temperatures will literally gel. Well, if that happens to a lipid bilayer of a cell, that's not compatible with life. Active transport is going to stop. Proton motive force is not going to be able to be formed. So probably what's happening at these minimum temperatures is that the particular chemistry of that membrane doesn't allow it to be fluid at that temperature. We'll talk a little bit about that before or in a bit. At high temperatures, notice that the optimum temperature is never very far away from the maximum temperature. Most bacterial species, once you're at the optimal temperature, you can't go up more than 5 or 10 degrees. And then suddenly, boom, the cells are going to just die. They're not going to be able to reproduce. This is also very easy to visualize because you all understand how proteins denature at high temperatures. So again, the example everyone always uses is the egg put into a frying pan, and you see the albumin denature and turn from clear to white. You see that denaturation. Well, that occurs not far above the optimum temperature in most bacteria, where certain, maybe not all of the proteins, but certain critical proteins will denature just a few degrees above the optimum temperature. And as always, humans, microbiologists, being humans for the most part, like to categorize microbes. So we lump them all into these various classifications, I think only to make students memorize them so that they can put them back on an exam. But if you think about these microbes, how they're going to behave at different temperatures, some of these are critically important ecologically. So things like the psychrophiles. So psychrophiles, you can tell from the name, are bacteria that love cold temperature. These are not only bacteria, but archaea as well. There are a lot of archaea, despite the fact that I've told you several times that archaea can be hyperthermophilic. They can love these really high temperatures. There are also archaea that do very, very well as psychrophiles. So these have optimal growth temperatures of less than 15 degrees centigrade. But notice where their maximum is, below 20 degrees centigrade. So I could challenge any of you in the microbiology lab, and I know that you would fail at this, if I asked you to go anywhere on this campus and find me a saprophytic bacteria growing in the environment. You couldn't do it because here, in this particular temperate environment, our temperatures always rise above, at some point, 18 degrees centigrade. So these microbes wouldn't survive. But you wouldn't have to go as far as you might think to find these microbes. You wouldn't have to go to the Arctic or the Antarctic to necessarily find them, because while you might think that saprophiles are ecologically very restricted to like the poles of the Earth, think about the oceans. The majority of the oceans at depth, especially once you get off the continental shelf where the ocean can get really deep, usually stays pretty consistently around 3 to 4 degrees centigrade. So in terms of just available space for microbes to grow, saprophiles probably have the largest ecological available niche of any of the microbes. So these are really fascinating microbes because you can't even bring them into the laboratory and grow them. Because, whereas you and I think about proteins denaturing in a frying pan, or maybe on a hot sidewalk in the summer when somebody says, oh, you could fry an egg on the sidewalk, it's so hot. That's when you and I think about proteins denaturing. These bacteria, their proteins will actually go through that same denaturation at temperatures lower than this. So as you bring them up to about 18 degrees centigrade, probably most of the proteins will just fall apart. They'll just lose their secondary or tertiary structures. They've evolved to work very well at low temperatures. The psychrotrophs, so moving up in temperature, the next class, are really an important class, largely because of the fact that, so if you look at their temperature optimum, it's around 20 to 30 degrees. But notice this cold tail. So the minimum temperature here drops off down, so that bacteria are still growing at 10 degrees centigrade, at 4 degrees centigrade, down even below 0 degrees centigrade. This is important because this classification contains most of the bacteria that are important in food spoilage. If you think about the fact that food still spoils in your refrigerator in the dorm or at home, it's because there are certain bacteria that, while that's not optimal for them to grow in your milk carton, they'll still do it, even at refrigerator temperatures, and they'll spoil that food. So psychrotrophs are important from a food spoilage point of view. Mesophiles, again, the next group moving to the left, moving to warmer climes, are all the bacteria that you know and love, that you're working with in the laboratory that will be your unknowns for the lab. These are microbes that have optimum around 55 to 65 degrees. No, I'm sorry, 20 to 45 degrees. I got ahead of myself. 20 to 45 degrees centigrade. So these are microbes that include all of the pathogens that you'll ever deal with. They all fall into the mesophiles. The thermophiles, I touched on those a little bit when I went off on Thermus aquaticus. Thermus aquaticus, that bacterium isolated in Yellowstone National Park, is a thermophile. It has an optimal temperature, actually a little bit higher than this, at about 72 degrees, but thermophiles have optimal temperatures between 55 and 65. Once you get warmer than this, once you move into the hyperthermophiles, there aren't even bacteria, for the most part, that survive much higher than that. Once you get into the classification of prokaryotic organisms that we refer to as hyperthermophiles, that have an optimum temperature 80 degrees or higher centigrade, almost exclusively archaea. Not even Thermus aquaticus, that thermophilic bacteria can do this. These are the archaea. They dominate it at that particular temperature. So what are some of the adaptations to these different temperatures that allow bacteria to survive and thrive at these temperatures? So we'll start out cold. We'll start with the psychrophiles. So as I mentioned, probably more than 90% of ocean water throughout the world is less than 5 degrees centigrade. So this is a huge domain where saprophytic bacteria really dominate. Bacteria that are mesophiles, thermophiles, with the exception of hot springs, like the black smokers that we talked about with Riftia. With the exception of those particular areas, most bacteria that live at depth in the ocean need to be saprophytic bacteria. And they're adapted to that by using different proportions of amino acids and different secondary structures to their amino acids to allow them to function at temperatures even below freezing. So the enzymes, will actually denature at moderate temperatures, as I mentioned before, 12 to 15 degrees centigrade, which is significantly cooler than this room is right now. These proteins will denature. Part of the reason why these proteins can function at low temperatures is they have much more alpha helix in terms of their character. Alpha helix, as opposed to beta sheet, alpha helix is much more flexible. And if you think about how enzymes work, whether it's a biosynthetic enzyme or a hydrolytic enzyme, it needs to be able to move. It needs to be able to physically change its conformation in order to accomplish that enzymatic activity. If that protein can't move at low temperatures because of a lot of beta sheets, it's not going to be functional. So evolutionarily over time, these microbes have changed the analogous proteins. So they still have DNA polymerase just like us. They still have RNA polymerase. Well, that's not just like us, but like other bacteria. And it's surprisingly not that different. But when you look at it, you'll see that it has evolved to have more alpha helix. So it's changed a few amino acids. Still does the same thing, but very beautifully adapted to low temperatures. Other adaptations that psychrophilic bacteria, they have an increased percentage of hydrophilic amino acids, water-loving amino acids, and thus a decreased percentage of hydrophobic amino acids. This concept is going to be a little bit easier to explain when I'm talking about the adaptation of thermophiles and hyperthermophiles, because bacteria and archaea that live at very high temperatures have the opposite adaptation. They have an increased percentage of hydrophobic amino acids. And when we get to that point, I'll explain it there because I think it'll make more sense when we talk about how an increased percentage of hydrophobic amino acids will stabilize proteins at high temperature rather than the converse. But for the moment, you just want to leave that thought in terms of how psychrophilic enzymes have adapted. But one of the major adaptations that psychrophiles must have is adaptations in the membrane. Because I mentioned that the typical membranes like yours and mine would actually congeal at temperatures such as this if we weren't able to regulate our body temperature. So the psychrophilic bacteria actually have an increased amount of unsaturated fatty acids. So these are fatty acids that will not gel at low temperature as easily. A lot of the fatty acids are even polyunsaturated fatty acids. So just to refresh people's memories and what I'm talking about by unsaturation units, here's a typical phospholipid. Here is the phosphate group. Here's the glycerol head with the ester linkages to the fatty acids. Now, here, there's a double bond in that fatty acid. This is an unsaturation unit. So this has one unsaturation unit. You can see that there's a kink that's put in that fatty acid because of that double bond. What this does is this tends to push the fatty acids a little bit further apart. Because remember that the van der Waals forces that are holding this together, if this is all saturated fatty acids, as temperatures get lower and lower, those molecules get closer and closer together, that the van der Waals forces can actually collapse that enzyme. They can become strong enough, even though they're very weak forces, they can become strong enough when the temperature gets low enough that this membrane shown on the top with the saturated fatty acids will actually gel. It will collapse. But if, on the other hand, the psychrophilic bacteria has put in unsaturated or even polyunsaturated, so multiple kinks in these fatty acids, this tends to push the members of that lipid bilayer further apart, so that as they come closer and closer together at lower temperatures, there's less chance that they can collapse, that they can gel. So these types of membranes with unsaturated fatty acids are much better adapted to cold temperatures. So in the last second, I just want to begin to introduce adaptations of thermo and hyperthermophilic microbes. And really, the enzymes of mesophiles, bacteria that like temperatures like yours and mine, denature at about 56 degrees centigrade, most of them. Those of the thermo and hyperthermophilic work optimally at between 55 and 113 degrees centigrade. So the same temperatures that would fry an egg, that would cause albumin to denature and come out of fluid, that's actually where these enzymes work best. So when we come back on Wednesday, I wish it was going to be Friday, we'll talk about some of the molecular adaptations of the enzymes in the hyperthermophiles, because from an industrial point of view, a biotechnological point of view, this is a really critical aspect. So we'll pick this up again on Wednesday.